So let's look at the tertiary structure of a protein. This refers to the three-dimensional folding of a protein. So the parts of a tertiary structure of a protein involve the beta-pleated sheet, the motifs, the alpha helices, and the random coil conformations. So I have drawn here a structure of a tertiary structure of a protein. And this one involves the random coil conformation, the beta turn, the beta pleated sheet, and also the alpha helix. Another thing that you are supposed to know is that the tertiary structure of a protein is stabilized by non-covalent bonds. So these bonds include the hydrophobic interactions. Side chains of neutral amino acids tend to associate with each other. Number 2 is electrostatic bonds. So this refers to the interaction between oppositely charged groups. For example, the amino end of lysine is positively charged, while the carboxyl group of aspartate is negatively charged at a physiological pH. So these oppositely charged groups tend to interact with each other. So through this interaction, they stabilize the protein structure. Number 3 is the hydrogen bonding. So a hydrogen bond refers to the weak ionic interactions between a positively charged hydrogen ion and a group of atoms that are negatively charged. So this group of atoms are normally found in the side chains and also on the carboxyl group where we have our oxygen atom that's negatively charged. On the side chains, we normally have nitrogen atom. We also have sulfur atoms. So these weak ionic interactions constitute the hydrogen bond. So this hydrogen bond helps to stabilize the protein tertiary structure. Lastly is the van der Waals forces of attraction. This refers to the interaction between uncharged groups of a protein molecule. This one also helps to maintain a protein tertiary structure. So we go through our notes. I've said that the tertiary structure refers to three-dimensional folding of a polypeptide, which consists of alpha helices, beta pleated sheets, beta turns, motifs, and random coil conformations. So I have drawn the structure. We have alpha helices. In alpha helices, we said that this structure normally coils along a given axis. So you can identify an alpha helices. Then we have a beta pleated sheet. The main feature of beta pleated sheet, we say that it is highly organized into sheet-like structures with the side chains, that is the R groups appearing either above or below the plane of the sheet. Then here we have a beta turn. We've just discussed the beta turns, and we say that each beta turn has got 4 amino acid residues, where the features that I've just described there, I said that the first and the fourth amino acids form a bond, and whereas the central amino acids do not form any interactions, they do not exhibit any interactions. Then we have random coil conformation. We said that at a random coil conformation, there is no order. That is a disordered conformation where we do not have alpha helices or beta pleated sheets nor beta turns. So that one is known as random coil conformation. I have also shown an amino group. This is the terminal end of a given protein. Then the tertiary structure of a protein is stabilized by non-covalent bonds. So these non-covalent bonds include the hydrophobic interactions. That is the first one. The non-polar side chains of neutral amino acids tend to associate in proteins. The process is called hydrophobic interactions, which help maintain the tertiary structure. Next is electrostatic bonds. These are bonds formed between oppositely charged groups of amino acid side chains. For example, the terminal amino groups of lysine is positively charged, and the non-alpha carboxyl group of aspartic acid is negatively charged at a physiological pH. They interact electrostatically to stabilize the tertiary structure. Next is the internal hydrogen bonds. Amino acids are involved in internal hydrogen bond formation, e.g., hydroxyl group of serine and threonine, the carbonyl oxygen of glutamine and asparagine, and the ring nitrogen of histidine participate in internal hydrogen bond formation. I have deleted this because it is the repetition of participation. Then van der Waals interactions. It refers to weak interactions between uncharged groups of a protein molecule. They also contribute to stability of proteins.